All right, guys, so today we're going to change gears and we finished the chemistry chapter, and now we're going to move on to the chapter on the cell. As always, the chapter begins with a list of objectives. These are the things that you should utilize as you study. Make sure that you can answer or describe each of these in great detail. There's more. All right. So cells are the smallest living units that can perform all vital functions or all functions necessary for life. And living things are made of cells. There are some living organisms that are what we call unicellular, meaning that they are just one single cell. And then there are organisms that are made up of trillions of cells. That's like the human. Humans are made out of 75 trillion cells. Each of these cells is specialized to perform specific functions for the body. So we're made out of 75 trillion cells, and cells are the building blocks of all living organisms, but there are all different types of cells, and different types of cells look a little bit different from each other, and they do different things. Just as we see with all other parts of the body, with cells, the structure of the cell is going to be related to the cell's function. For example, muscle cells. Muscle cells are filled with proteins. When you look inside them, they're almost all protein. And those proteins are used for contraction. So that's why that cell is full of contractile proteins, because its function is to contract. Skin cells don't look like that. Skin cells don't have all of those proteins because they're not made to contract. So we have all different types of cells. Epithelial cells, which are skin cells. We have cardiac muscle cells in the heart, skeletal muscle cells. We have osteocytes, which are bone cells. And each of these has their own special function that allows them to do what they need to do to keep the body working, to keep the body healthy. But all of these cells are living. All of these cells have the ability to maintain their own homeostasis or balance. We're going to talk about some of the general features that most cells have. So by the end of this chapter, you should have a really good idea about the overall structure of a cell and the different parts of a cell and the way that a cell works. Then as we go through the rest of the semester, we'll look at some very specific types of cells, and I'll point out some of the differences between them or some of the special functions that they have, based on where they are in the body or what part of the organ or what organ they're a part of. So we'll start by talking about the plasma membrane. The plasma membrane or cell membrane is the outer wall, the wall around the entire outside of the cell that separates the cell from its environment. So we see that the plasma membrane is this outer barrier around the cell. So it's like the wall around the outside of your house. It keeps all the good stuff in, keeps all the bad stuff out. Now, if we were going to be technical, instead of saying stuff inside and stuff outside, we would say cytoplasm and extracellular fluid. So the stuff that's inside the cell, technically, and outside the nucleus, which we'll mention in a little while. So this is like the nucleus inside the cell. All of this inside the cell is the cytoplasm. The extracellular fluid refers to all of this fluid that's outside the cell. Most of our cells are surrounded by water, right? Our body's full of water. They're surrounded by fluid, and that's extracellular fluid. Think X is like outside, like when you exit, you leave. External is the outside. So extracellular is outside the cell. This extracellular fluid we also call interstitial fluid. So you'll hear me say interstitial fluid or IF a lot. All of our cells are surrounded by interstitial fluid. So interstitial fluid is the fluid that surrounds our cells. Again, the cytoplasm refers to the fluid inside the cell. When we look at the cytoplasm really has two components. Cytosol, that's just the fluid, and then organelles. So all of this cytoplasm inside the cell is organelles, right, spread throughout a fluid, an intracellular fluid. Now, organelles literally means tiny organs. And organelles are literally like organs, but for the cell. Just like in your body, you have all different organs. They're shaped a little different from each other and they do different things, but they keep your body working. The organelles are a group of structures inside the cell that keep the cell working. They're all a little bit different from each other. They're shaped different from each other. They have their own separate functions, but they all keep the cell working and running. We're going to look at a bunch of different types of organelles as we go through this chapter. Intracellular means inside the cell. Just like extracellular is outside the cell, intracellular is inside the cell. So the functions of the plasma membrane. The most obvious function of the plasma membrane is that it's a barrier, right? It's a wall around the outside of the cell that provides physical isolation, right? It keeps stuff out of the cell that doesn't need to come in, and then it holds all of the organelles and the fluid inside. Now, this barrier, this wall is great because it provides protection, but we don't want it to be impenetrable, right? We don't want it to be a complete absolute wall that doesn't let anything through. Think about your house. Again, we said that the wall around the very outside of your house is great. It protects you. It keeps all of your stuff inside. It keeps other people outside. But imagine if that wall around your house had no doors and no windows. Instead of protecting you, that would hurt you, right? You can't survive if there's no way for anything to get in and out of your house. And the cell is the same way. The plasma membrane provides a barrier for physical isolation, and that's great. That provides protection for the cell. But there's got to be some way to get stuff in and out of the cell. And the plasma membrane does that. It has proteins and channels present that allow the plasma membrane to regulate or control the exchange of things with the environment. So in other words, it controls what enters and leaves the cell. Just like, again, you have doors and windows. You can open the door when you want to bring groceries in. You can open the window when you want to let air in or out. You can open the door to take your garbage out. Well, the cell does all of this stuff as well. The cell can open these doors to allow ions or nutrients, like food, right, glucose, to enter. It can also open these doors to allow waste products or helpful things that the cell is making, like hormones, for example, to leave the cell. So the plasma membrane provides protection and isolation, but it also allows the cell to control what gets in and what gets out. The plasma membrane provides sensitivity to the environment, so the cell knows what's going on around it and it can receive messages from other cells. There are little receptors that are present on the plasma membrane, and these receptors receive chemical signals. So chemical signals bind to the receptors and they give that cell information. The plasma membrane also allows for extra structural support of the cell. There are proteins that stick in and out of the plasma membrane, and those allow the cells to be anchored to each other. So two cells can be connected to each other. or the cell can be connected to another tissue. It allows the cells to have extra structural support, extra strength. The plasma membrane is made mostly out of a phospholipid bilayer. You guys should recognize this term, phospholipid. Right? Remember, we looked at those when we looked at organic compounds. They're a specific type of lipid. Bilayer, bi meaning two, like a bicycle has two wheels. So 2 layers of phospholipids. And this is what you see right here. The phospholipids are the little structures that have the heads and then the little tails coming out. And you see they're arranged in 2 layers. They have their heads like this towards the outside of the cell, and then their tails go inside the membrane. And then the other layer has the heads towards the inside of the cell, and their tails also go inside the membrane. Remember that phospholipids are special because the head and the tail behave completely differently from each other. The head region is hydrophilic. Remember, that means it likes water. It interacts with water perfectly fine. So those hydrophilic head regions are located like towards each side of the membrane because there's water on each side, right? Out here, there's the extracellular fluid, and in here is the intracellular fluid, right? The cytoplasm. So the parts of these phospholipids that like water are on the outsides of the membrane where all of the water is. The tail region is hydrophobic. And remember, a phobia is a fear. So the tail region is water fearing. It does not like water, right? It's really lipidy or fatty. So it tries to get away from the water that's both outside and inside the cell. That's why the tails are inside the plasma membrane. They're in this region here where they can try and get away from the water. Now, this works great as a barrier to anything that likes water. So remember, water is a polar compound. Remember when we looked at it, it had parts that were kind of positive and parts that were kind of negative. And we use this saying, likes dissolve likes. This means that like compounds will interact with each other. And since water is polar, it kind of has charges. That means that other things with charges will interact with the water. They're perfectly fine. But things that are non-polar, things that don't have charges, will not interact with the water, okay? Because they're different. So this plasma membrane with this whole lipidy center right here acts as a barrier to anything that likes water. So things like ions. Remember, ions are just charged atoms. So sodium, chloride, potassium, calcium, those all have charges. So they will not simply cross through this plasma membrane because they don't want to come in contact with those lipids in the middle. So they just won't do it. The same thing goes with other larger compounds that are polar, like water. So water or glucose, for example, has slight charges to it. So it will not simply cross through the plasma membrane because it doesn't want to go through all of those lipids. It doesn't like lipids. Things that do like lipids, right? So stuff that's also hydrophobic, stuff that likes lipids, that is non-polar, will go straight through that plasma membrane. So examples of that include fatty acids, right? Remember fatty acids, those long tails, they'll go straight through because they don't mind those lipids. The gases that we breathe, oxygen and CO2, those are lipophilic. They like lipids, so they'll go straight through the plasma membrane. but the fatty area in the middle acts as a barrier to anything that's water soluble, that has any charge. So anything that's charged or polar, it will not cross straight through those phospholipids. Now, the plasma membrane is made out of mostly a phospholipid bilayer, but it does have proteins scattered throughout it. And those proteins are really what give the membrane all of those different functions, like allowing things to cross the plasma membrane or receiving signals. All of those special functions happen because of the membrane proteins that we see scattered throughout the plasma membrane. There are anchoring proteins. These are just the stabilizers. They can go inside the cell and attach to the cell's skeleton, or they can stick outside the cell and anchor multiple cells to each other. They're just the proteins that provide stabilization and strength for the cell. There are what we call recognition proteins that stick out of the plasma membrane. All cells in our body have these. They're referred to as antigens, but they're just like little ID cards. So we have, our cell, and it's got these little IDs that stick off of it, and they just identify this cell as one of our normal body cells. Our immune system will go through our body and it checks all of these IDs. Right? And if it's normal, one of our normal body cells, the immune system ignores it. It's fine, it's supposed to be there. But if this ID card is not normal, say it's a bacteria, well, the bacteria is going to have an ID that doesn't match our body. So in that case, the immune system would destroy it. Or say this is one of our cells that's been infected by a virus or it's sick in some way, then its ID card's not going to be normal anymore. And then the immune system would destroy it. So we have proteins that stick off of the plasma membrane that act as identifiers or recognition proteins, and they're called antigens. There are some proteins that are present in the plasma membrane that act as enzymes. Remember, enzymes just catalyze chemical reactions or speed up chemical reactions. They can speed up chemical reactions that are happening here inside the cell or outside the cell, just depending on which way they stick out of the plasma membrane. There are receptor proteins present. Remember, I said that receptors bind to something and then they make something happen in the cell. This is how the cell receives messages and instructions from other areas of the body. So receptor proteins bind to and respond to a ligand. This word ligand is just anything that binds to a receptor. It could be an ion that binds to it. It could be a hormone that binds to it. It could be a neurotransmitter that binds. The point is that these receptor proteins allow the cell to get messages from other cells. For example, insulin is a hormone that binds to a receptor protein, and then that allows the cell to absorb glucose. Right? So we have this plasma membrane here, and there's a little channel, a little protein that will allow glucose to enter into the cell. But this channel, this little protein is closed, it's turned off, so it won't allow the glucose in. But there's a little receptor that's associated with it, and that receptor binds to insulin. So when insulin binds to this receptor right here, that opens up this channel, and then the glucose is allowed to enter. Okay, so the only reason the glucose was allowed to enter is because this insulin was able to bind to its receptor. So the binding of insulin to the receptor opened up this channel to allow glucose into the cell. Okay, so the cell was able to receive a message. It was able to receive that insulin message because of a receptor protein on the plasma membrane. There are also carrier proteins and channels or pores. Both of these just allow things to enter and leave the cell. Remember, we said that there are a lot of things that will not go straight through the plasma membrane, right? Anything that's charged in any way, so ions, polar compounds, or things that are really big, they can't cross straight through the plasma membrane. So we need another way to get them in and out of our cells. We can do that via carrier proteins, which will grab a hold of something and physically move it across the plasma membrane. Or channels or pores, which are a little bit smaller, and they're just like they sound, they're channels. They're like little tunnels that are made out of protein that allow things in and out of the cell. We'll talk a lot more about these and the way that they work and the types of things that they transport. We'll talk about whether or not they require ATP or energy. All of that will be in probably the third lecture that we do on the cell. All right, so we said that the plasma membrane is the outer barrier around the cell. Deep in our cell, in the very center of our cell here, we have this really dense core called the nucleus. Just like in the atom, remember the nucleus was the center of the atom? The center of the cell is the nucleus as well. The nucleus is where we have our DNA in this nice protected, safe environment in the middle of our cell. So we have this plasma membrane around the outside, we have the nucleus in the center. Everything in between is referred to as the cytoplasm. So all of the materials inside the plasma membrane and outside the nucleus are included in the cytoplasm. And I told you guys that cytoplasm has two components, right? The cytosol and the organelles. The cytosol is the fluid part, the liquid. We refer to it as intracellular fluid because it's the fluid within the cell. This fluid has some stuff dissolved in it, right? It's not just plain water. It has some nutrients in there, right? Like glucose and amino acids, right? Some fatty acids as well. It has ions present, hey, like potassium, for example. It has some proteins and then some waste products that need to leave the cell. So it's not just plain water, right? It's thicker than that. There's some stuff dissolved in the water. Now, when we look at this intracellular fluid, it does not match extracellular fluid. So the fluid inside the cell is different from the fluid outside the cell. Inside the cell, there is high potassium. So there's more potassium inside than there is outside. And then the others are opposite. There's less sodium, so there's more sodium out here, less calcium. That means there's more calcium outside, and chloride. This is what the general cell looks like up here. Hey, more potassium inside, more of everything else outside. There's also more proteins inside the cell, and proteins happen to have negative charges. And because there are so many negatively charged proteins inside the cell, we see that overall, the overall charge inside our cell happens to be negative compared to outside. Okay, all of those are things, like these facts, are going to be important later, especially when we talk about muscles and how muscles work. And then if you go into A&P, that concept is going to be important for a ton of different chapters. So it's really important to know this, that the plasma membrane, because it's a barrier to ions and it stops ions from moving in and out, it's able to keep them kind of separate from each other, and it's able to keep more potassium inside, and it's able to keep the sodium, calcium, and chloride outside. Okay? And then the fact that it's more negative inside the cell compared to outside the cell is also important. Okay, so the cytoplasm is everything inside the plasma membrane but outside the nucleus. And cytoplasm contains cytosol, which is the liquid and then all the stuff dissolved in it, and then organelles. And I said that organelles are tiny organs. They're little functional structures that are present in the cell that keep the cell functioning and keep the cell healthy. Now, we're going to talk about all of these organelles. I will warn you, this list goes on and on and on, and it will feel like it never ends. We're going to go through most of the organelles today, or a lot of them today, and then in the next lecture, we'll finish them up. But just bear with me. It does end up being important, and they do kind of come back at different times throughout the semester. First, we'll start with the cytoskeleton. Cyto means cell. So cytoskeleton is literally telling us the cell's skeleton. This is composed of a bunch of just strong fibers, strong proteins called microtubules, intermediate filaments, microfilaments. Again, these are all just structural proteins. And the cytoskeleton gives the cell shape and strength, just like our skeleton. Right? Our skeleton, or all our bones, give us our general shape and stature and size and strength. They keep us strong and provide protection. The cytoskeleton also enables movement of some other organelles. Part of the cytoskeleton kind of radiates out around the cell like this, kind of like the spokes on a wheel. Like if you look at the bicycle wheel, how the thin spokes, the wires, radiate out from the center and they give strength. Well, our cytoskeleton kind of radiates out like that. And these little spokes, these tubules, act as a railway system. And organelles, some of the other little structures, can latch on and can move down these spokes. So they can like move from one area of the cell to another along this mini railway system. So that's kind of in general the cytoskeleton. Microvilli are little finger-like protrusions. So little finger-like protrusions of the plasma membrane. So instead of being flat on the surface, right, or rounded on the surface, the plasma membrane goes up and down, up and down, up and down like this. Right, so the surface of the cell would look like that. The reason that we have microvilli are that they increase the surface area of the cell. So like there's more space with this curvy line than there would be if it was just a straight line. This is important in areas where we absorb things. So specifically in the GI tract, right, the small intestine, where we absorb most of the nutrients from our GI tract into the body. Okay, there are little like transporters or little pumps that are present here on the plasma membrane. And they take nutrients from the GI tract and they get absorbed into the cell and then into the body. So the fact that these microvilli, right, make the plasma membrane really curved means we can fit more of these transporters, which means we can absorb more nutrients. Okay, so microvilli are really important in the small intestine, part of the GI tract. These microvilli do happen to attach to the cytoskeleton. They have proteins in the center of them that anchor them to the cytoskeleton to give them strength so that it's not like all floppy and mushy. The next organelles that we'll talk about are centrioles. All animal cells that have the ability to divide, right? So one cell replicates and splits into two cells. All cells that can do that, that can divide, have a pair of centrioles. Okay, they look like this. They look like these two little cylinders that are like at right angles to each other. You see one of them's coming this way and then the other one's going the other way. And they sit right next to the nucleus of the cell. So if you look at this cell up here, the nucleus is in the middle, and then you see the centrioles sit right here, right next to it. Each centriole is made of 9 microtubule triplets. So they're made out of microtubules, which are just little proteins, and they look like little tubes. So this is like one triplet. A triplet means 3. So there's 3 tubules to make that triplet. And then there's 3 tubes to make this triplet, right? 3 tubes to make this triplet. So there are 9 of those triplets that all go around in a circle to make this cylinder. Now, I told you guys that these centrioles are important in cell division. Specifically, what they do is they form something called the spindle apparatus. And what this does is it separates the DNA into 2 equal parts when the cell is dividing. So if I have this one cell and it's going to end up splitting off, right? It's going to start splitting like that, and eventually it'll end up as 2 separate cells. When the cell divides, what happens is the first thing is it replicates its DNA, because at the very end, both cells need a full copy of DNA. So the cell is going to copy his DNA. So he has 2 sets. And then that DNA lines up in the very middle of the cell like this. And you have, one set on one side, one set on the other. Well, what the spindle apparatus does is the centrioles attach these spindles to the DNA, and the DNA splits down the middle. And then the centrioles pull their copy of DNA over to each side. So what you end up with is your centriole with, your one set of DNA, and then your centriole with the other set of DNA. So the cell can split, and you know that the DNA was divided equally. We'll talk more about how cell division works at the very end of this chapter. But for right now, you guys should know that these are centrioles. They're made out of microtubules, and they form the spindle apparatus during cell division to separate the DNA. Cilia and flagella are kind of similar to each other structurally. They're both also made of little microtubules that stick off of the cell's surface. But they are different. They're both small, little, thin, hair-like projections from the cell, and they can both move. Both the cilia and flagella can whip or move themselves. But that's where the similarity ends. They have very different functions. When we look at cilia are numerous short projections. So there are tons of cilia that stick off of a surface of the cell. We happen to have them in the trachea. Really, we have them lining most of the respiratory tract, right, that brings air down into our lungs. So we have these tubes that bring air down into our lungs like this. And the cells that line these tubes have tons of cilia on their surface. So tons of these little hairs that stick off like this. When you look at it under the microscope, it actually looks like shag carpet. So when you look at a shag carpet, how you see all the little hairs, all the little fibers, that's what it looks like. So there are tons of them. And what these cilia do are they move rhythmically all together. So they'll bend, push, and that allows them to move fluids like mucus across the cell's surface. Now, the reason this is important in the respiratory tract is that we don't want mucus to drip down into our lungs. We coat the surface of the respiratory tract with mucus because it's sticky and it traps all the nasty stuff that we breathe in. But we don't want that nasty stuff to get trapped in the mucus and then have the mucus drip down into our lungs, which is what gravity would do. Instead, what happens is the mucus traps the bad stuff, and then the cilia move and push the mucus up so we get rid of it. And once it gets to your throat, you feel that tickle and you pull the mucus up. You can spit it out, you can swallow it, but either way, it's not going into your lungs. So the cilia move to push the mucus up and away from the lungs. Now, a flagellum is one single long projection that moves the cell forward. So cilia move stuff across the cell, but the cell stays where it is. Flagellum has this one long tail, so you have like the cell, and then there's one long, like kind of whip-like tail that extends from it. And that tail is the flagellum. And again, this tail moves. It moves in kind of like a corkscrew motion, and that propels the cell. So it makes it look like the cell is able to kind of swim, to move from place to place. The only cell in the human body that has a flagellum is a sperm cell. Okay. Now, once the sperm get ejaculated into the vagina, they have to swim. They have to enter into the uterus and into the uterine tubes, and then they have to enter into the ovum or egg in order for reproduction to occur. That's a long distance for a microscopic little cell. That takes a lot of energy, a lot of swimming. So the flagellum is important for the sperm to be able to accomplish that. Now, flagella, that's plural, like when I'm talking about many of them. A flagellum is just one. That's why you keep hearing me say flagellum. Flagellum means just one tail. Okay, the next organelle that we'll talk about is the ribosome. Ribosomes are made out of both RNA, specifically rRNA, which means ribosomal RNA, and protein. And ribosomes are important because they're the initial site of protein synthesis. This is specifically where amino acids are connected to form a polypeptide. So remember in the chemistry chapter, we looked at proteins, and we said that proteins were these long chains of amino acids. Making a protein takes a lot. There are a lot of different organelles that are involved. But the first thing that we have to do is connect all of these amino acids to each other. That happens at the ribosome. Hey, the ribosome reads the recipe on the RNA and it links the amino acids together. So it makes this polypeptide, this chain of amino acids. Now there are two types of ribosomes, free ribosomes and fixed ribosomes. Free ribosomes are free floating in the cytoplasm. They're not actually anchored to anything. They typically will manufacture proteins for that cell. So they'll string together the amino acids and then they'll just release the polypeptide into the cell. Fixed ribosomes are fixed. They're anchored or attached to the endoplasmic reticulum, which is another organelle that we'll look at. Fixed ribosomes will make the polypeptide and then they'll push it into the endoplasmic reticulum, and the endoplasmic reticulum will process it further. The fixed ribosomes can be used to make proteins for that cell, but it's also used to make proteins for secretion. So proteins that are going to end up getting pushed out of the cell to go, somewhere else in the body to do something. Here we'll talk about that endoplasmic reticulum that I just mentioned. So the endoplasmic reticulum is this membrane enclosed structure that extends off from the nucleus. So if you look at the cell, you'll see the nucleus in the middle, and then this like membrane bound structure that comes off of it like this. And that's the endoplasmic reticulum. Now, there are like two parts to the endoplasmic reticulum, and they almost act like two different structures. They're different from each other, but they are interconnected. It literally flows from the rough endoplasmic reticulum into the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, or smooth ER, is called smooth because its surface is smooth. It doesn't have any ribosomes attached to its surface. So there are no ribosomes attached to it, means it's not involved in protein synthesis. Instead, what the smooth ER does is synthesizes lipids. So this is where we make lipids, so where we make cholesterol, where we make steroids. They're made in the smooth ER. This is also where in specific cells we store huge amounts of calcium. Muscle cells have a very specialized smooth ER that stores a huge concentration of calcium. But we'll talk about that more when we look at the actual muscle chapter. The rough endoplasmic reticulum, or rough ER, it's called rough because it's rough. Its surface is really bumpy because the surface is covered with ribosomes. Remember, we just said that there are free ribosomes that just float around the cytoplasm, and then there are fixed ribosomes that are attached to the endoplasmic reticulum. Okay, so the part of the endoplasmic reticulum that has the ribosomes attached is called the rough ER because it's literally a bumpy surface. The rough endoplasmic reticulum is active in protein modification and glycoprotein synthesis. Remember that these ribosomes on the surface are going to start making proteins, right? They connect a bunch of amino acids and then they shove that polypeptide into the rough ER. Well, the rough ER takes that polypeptide and does stuff to it. It can, cut off the ends or connect it with something else and start to help it fold into its shape. It can just make little changes to help the protein get to where it's eventually going to be, to help it get to its final form. A glycoprotein is just when there's a sugar attached to the protein. So it can do that. It can take a sugar and attach it to the protein. But the point is, because it's covered with ribosomes and the ribosomes push the polypeptide into it, they then modify that polypeptide to help make a protein. After the endoplasmic reticulum is done with that protein, it encloses it in a transport vesicle. So it pinches off of the rough ER in a little bubble. And that transport vesicle normally takes the protein to the Golgi apparatus, or Golgi complex, which is another organelle. All right, so here you guys see the endoplasmic reticulum. Again, it like folds off of the nucleus of the cell, and you can see that this first part here, right, this first part is the rough endoplasmic reticulum because it's got these ribosomes all over the surface. And then eventually that kind of folds out into the smooth endoplasmic reticulum that does not have any ribosomes on its surface. This part over here, right, the rough ER, that's the part where we're going to be modifying proteins, helping in protein synthesis. This part over here is where we're going to be making lipids. Let's go ahead and do the Golgi apparatus, and then I'll stop after that. The Golgi apparatus, or it's also called the Golgi complex, is an organelle that it also has its own membrane around it. And the membrane kind of looks like a series of like stacked dinner plates or stacked pancakes. So there's stacked disc-like cisterna. Cisterna is just like a chamber, a storage chamber. So it kind of goes like this. And it's like a stack of plates or a stack of pancakes. The Golgi complex is important in carbohydrate synthesis. So making sugars occurs in the Golgi complex or Golgi apparatus. It also puts the very finishing touches on proteins. I just told you guys that when the protein leaves the rough ER, it typically comes to the Golgi. And the Golgi will do the last little finishing touches on the protein. Like maybe take a few tertiary structures and combine them to that final quaternary structure. Also, the Golgi apparatus acts as a packaging plant. So any protein that needs to be packaged for long-term storage or for secretion out of a cell gets packaged in the Golgi apparatus. So the two things I think of when I think of the Golgi are carbohydrate synthesis, so making carbs, and packaging plant. I cannot get enough water today. It's like constant. So the new proteins will like fuse, they'll enter into the Golgi apparatus. The Golgi will do whatever with them, and then they'll pinch off of the Golgi apparatus in these little vesicles. Right? Again, a vesicle is just like a little chamber. It's got a little membrane around it. And these proteins can be packaged for multiple reasons. For secretion out of the cell. So if you're making a protein that you then want to release from the cell, or for later use in the cell. Sometimes proteins are really reactive and you can't leave them just floating around the cell by themselves, like some enzymes, for example. You don't want just floating around the cell freely because they'll start to make things happen all over the place before you're ready. So you can package them in a vesicle and then use them later. One key example of this is that the Golgi apparatus packages a bunch of digestive enzymes inside a vesicle. Digestive as in like to digest something is to break it down. So all of these like nasty enzymes that'll chew things up and break things up get stored inside a vesicle. And that creates another organelle called the lysosome. So a lysosome is a vesicle containing digestive enzymes. Now, can you think about why we might put these enzymes inside a vesicle? Why wouldn't we just want them floating around the cell? Well, I just said they digest things. They break things down. So if we let them just float around the cell freely, they would actually digest the cell. They would break the cell down. So we package them in this little vesicle until we have something very specific that we want to fuse with it to digest. This is showing us the Golgi apparatus or Golgi complex. Here again, you see it's like this stack of plates. These would be transport vesicles from the endoplasmic reticulum, right, that are bringing proteins in. So these just like fuse, they melt into it and dump their proteins in. And then they get processed, right, and then the proteins eventually get pinched off. And then we have these new vesicles with the proteins in them. I'm going to stop there. The whole concept of producing ATP or producing cellular energy in the mitochondria is a little complex, but it's very, very important. It's a super important concept. So I want to start it when you guys are fresh and ready to go. So I'll go ahead and pause the lecture here, and then tomorrow we'll start off right here with mitochondria. If you guys have any if you have any questions, go ahead and shoot me an email. Otherwise, I hope you're all doing well and studying hard.